What you're looking at right here is the genetic code. And what's fascinating and unbelievable is just how simple it is. There are only 4 letters in the genetic code that spell out every amino acid of every protein in every living cell on the entire planet. All life uses this same genetic code with some rare exceptions, because evolution is that way. Whatever works can happen. But you use the code by reading it from the inside out. So, for example, if on messenger RNA the codon is AUG, you start with the A, then you come out to the U and come out to the G. And that's methionine. That's an amino acid. These are all amino acids. And remember that amino acids are what get strung together and bound together by peptide bonds into a polypeptide or a protein. That's what this is all about. So the genetic code starts in DNA. There are codons in DNA that get turned into codons in RNA, and then the codons in RNA get translated into the amino acids that go into making a protein. That's what we're looking at here. So the genetic code describes the way codons in DNA and messenger RNA, and I guess so again, there are codons in DNA that get transcribed into codons in messenger RNA, and then they get translated into amino acids during protein synthesis. And by the way, protein synthesis is the same thing as protein translation. Codons are nucleotide triplets. Not triplets in the sense that they're identical. They're not identical, obviously, like these are 3 different nucleotides here. So those triplets are in DNA, and again, they start in DNA, then they get transcribed into RNA. But they code for one specific amino acid. Each triplet, each codon, codes for one specific amino acid of the 20 that you'll find in the genetic code wheel here. Amino acids are the monomers of polypeptides or proteins, as you should know. I mean, we learned that in the last unit, the biochemistry unit. The relationship between codons and amino acids is not one-to-one. Some amino acids have more than one codon. So for especially like proline and arginine and threonine and valine and glycine and serine, there are 64 possible combinations of these 4 letters in codons. And so some of these, for example, proline here, CCU, CCC, CCA, CCG, all code for proline. That's what we call wobble in the genetic code. And it protects against mutations. If a mutation happens changing a codon from CCU to CCC, there's no change in the protein. Or if it mutates to CCA or CCG, there's no change in the protein. Proline still gets inserted where it is supposed to be inserted into the polypeptide chain. So this wobble, this redundancy in the genetic code protects against mutation. AUG codes for the amino acid methionine, but it's also the start codon, AUG, methionine. And it would be a good idea to write start right here. It's not only methionine, it's start. All proteins, the translation of all proteins begin with methionine. There are 3 stop codons. So here's one, UGA, and then there are 2 here, UAA and UAG. So there are 3 stop codons. The stop codons signify the termination of translation. Termination, the end, right, of translation. The end of making a polypeptide or a protein. And the end of the polypeptide being synthesized. The messenger RNA nucleotide sequence, hopefully you're getting the idea here that sequence is important in understanding all of what's going on in gene expression. The messenger RNA nucleotide sequence, that's what we're looking at right here, is read as a codon sequence, codon, right? A sequence of codons. Every 3 nucleotide is a codon. that is translated into an amino acid sequence of a polypeptide or protein. That's gene expression. That's most of gene expression. It's also the central dogma of biology. Okay, so let's review. Messenger RNA is made in the nucleoplasm. That's like the cytoplasm of the nucleus, right? The nucleoplasm, inside the nucleus, by transcription and processing. Remember that after RNA is transcribed, it is processed. The introns are cut out, the exons are spliced together, there's a cap and a tail placed onto that messenger RNA. Messenger RNA goes out into the cytoplasm and binds to a ribosome. We're not seeing that in this image, but that's what's going to happen next to this messenger RNA. Messenger RNA is able to leave the nucleus through nuclear pores due to it being a single polynucleotide. Unlike DNA, that's a double helix, right? Well, RNA is a single polynucleotide. That makes it about half the width of DNA. Therefore, it's better able to fit through the nuclear pores. DNA never leaves the nucleus. And the cap and tail that it receives during processing. There's a cap at the start end, the AUG end, and there's a tail at the other end that we can't see in this diagram. But that also helps it get through the nuclear pores. It's like a gate key, the cap. Messenger RNA participates in the process of translation. Translation is the making of proteins, putting polypeptides into a chain to form a polypeptide, putting amino acids into a chain to form a polypeptide, which can be a protein, or it will join up with other polypeptides to be a protein. So translation is one of the final steps in gene expression that produces the resulting phenotype. Following translation, the polypeptide or protein may be further modified or regulated. So that's also part of gene expression. But one of the major last steps in gene expression is protein translation, stringing amino acids together into a particular sequence to make a particular protein that has a particular function. So translation is the synthesis of polypeptides or proteins. ... meaning that we're going to take a look at that process next. How does that actually happen? Once we have the messenger RNA, how does the messenger RNA get translated into a polypeptide or protein? This is the same thing that you're working through in the lab, so hopefully you'll have a really good understanding of how this works from that. It's difficult to understand the process here because it is a dynamic process. It's moving. Components are interacting with each other and changing, and that's difficult to understand in a static image like this. So that's why you're working on the lab. That's why there are all kinds of videos that I would suggest you watch to see how this process actually works. But messenger RNA carries the nucleotide sequence of the gene to be translated, right? So here's the nucleotide sequence in messenger RNA. Transfer RNA, these guys, transfer. That's why they're called transfer RNA. They really could be called translate RNA, though, because they're the ones that actually do the translating between the language of nucleotide sequence and the language of amino acid sequence. And that's why this whole process is called translation, because we're going from one language to another, just like if you translate English into Spanish. So transfer RNA transfers an amino acid specific to its anticodon. Each transfer RNA has an anticodon, and the amino acid that it's carrying is specific to that anticodon, which matches up with a specific codon, which is complementary to the messenger RNA codon. Yeah. So ribosomal RNA, the big blue and tan thing here in the background, the ribosome is made up partially of ribosomal RNA. There are also 80 different proteins, as we learned previously, that go into making up that RNA. But the ribosomal RNA gives the ribosome the ability to bind. It provides binding sites for both messenger RNA, that's in the small subunit of the ribosome, and transfer RNA. And there are actually 3 binding sites in transfer RNA. The ribosome's not being shown wide enough here to have 3 binding sites, but there's the A site and the P site, and there would also be an E site for exit. This allows the transfer RNAs to interact. This allows all the RNAs to interact and perform their function during translation. Translation begins when the capped end of the messenger RNA binds between the ribosomal subunits. So here's the capped end. It's the capped end is the one with the start codon, AUG. The ribosome reads along the messenger RNA sequence until it finds that start codon. Next, transfer RNAs with anticodons complementary to the mRNA codons bind to the ribosome. And at the initiation of translation, there are 2 transfer RNAs that are going to bind, bringing along with them 2 amino acids, matching up their anticodons to the codons. And methionine is first. Notice, MET, methionine. ALA is alanine. Once a peptide bond forms between the amino acids, the ribosome moves the distance of 1 codon downstream. So these arrows are showing the movement of the ribosome. It's also known as translocation. The ribosome translocates downstream along the messenger RNA after a bond forms between the first 2 amino acids, after the peptide bond forms. ... So we continue on here. Next, the start transfer RNA exits as another transfer RNA carrying the amino acid specific to the next codon/anticodon binds the ribosome. So when the ribosome translocates, and now we have a ribosome that's wide enough for all 3 binding sites, transfer RNA binding sites. So we got the A and the P and the E, and the E stands for exit. So when the ribosome translocates 1 codon over, 1 codon downstream, the first transfer RNA, the one that matches up, that has the anticodon that matches up with the start codon, is no longer bound, and it leaves. But it leaves behind its methionine because it's bound to alanine. As the ribosome moves along the messenger RNA, the cycle of transfer RNA amino acid delivery and peptide bond formation continues, producing a growing polypeptide chain. So we've already seen basically the whole process. Transfer RNAs come in, they move from the A site to the P site, a peptide bond forms between the amino acid that was delivered previously and the amino acid that's being delivered, and then the ribosome translocates and the transfer RNAs leave. So transfer RNAs come and they deliver their amino acids. That amino acid gets added to the growing polypeptide chain, and we end up with a polypeptide or protein. Because remember, some proteins are made up of only 1 polypeptide, while others are made up of more than 1 polypeptide. That's why we're always saying polypeptide or protein, polypeptide/protein. The cycle stops when the ribosome reaches 1 of 3 stop codons. So you can look back on the circular genetic code table or wheel. UGA is one of those, and there it is, UGA. Notice that there's no transfer RNA that's binding to it, though. No transfer RNA binds. There are protein release factors that do. So once a stop codon is reached, these proteins bind to the ribosome and cause everything to come apart. Everything dissociates. Dissociate simply is a fancy word for come apart. So both ribosomal subunits come apart, messenger RNA unbinds from the ribosome, transfer RNA unbinds from the ribosome. The last transfer RNA releases its final amino acid, and we have a polypeptide chain or a protein. So the product is a polypeptide/protein composed of a very specific sequence of amino acids. That sequence of amino acids came from the sequence of nucleotides in messenger RNA, which came from the sequence of nucleotides in DNA. There are thousands, hundreds of thousands of different proteins in the human body, for example, and I'm sorry, there was a mistake there. And all of them have a different amino acid sequence. Different proteins have a different amino acid sequence. That's what makes them different. That gives them a different structure, which gives them a different function. So the DNA sequence leads to a messenger RNA sequence, which leads to the amino acid sequence. That's basically the central dogma of biology. But again, you got to say it. The central dogma of biology. You got to say it like that because it's so important. So one more time, because it's important, the central dogma of biology. This diagram represents that. The process of gene expression, mostly. Again, there can be some processing and regulation after the polypeptide or protein is made through translation. But most of gene expression is here. And it begins with the sequence of nucleotides in DNA, that's what we're seeing here, that's transcribed and processed into the sequence of nucleotides in messenger RNA. Then the sequence of nucleotides in messenger RNA is translated into the sequence of amino acids in a polypeptide or protein. And that's what we're seeing here. Messenger RNA is translated into the amino acid sequence in a polypeptide or protein. And it's the amino acid sequence of a polypeptide or protein that produces its conformation. Remember, conformation is that fancy word for shape. And it's the conformation that allows it to carry out a specific function. This is the relationship, and it's a big idea in biology, if you'll recall, the relationship between structure and function. The structure of the protein, the conformation of the protein, is what gives it its function. And the highly variable nature of proteins enables them to perform a wide variety of functions crucial to living systems, most notably enzymes. There are enzymes that are regulating every chemical reaction that's taking place as part of the metabolism in your body right now. DNA codes for proteins, and proteins run the show, as we say. Actually, I think I'm the only one that says that. Next, we'll take a look at what happens if something goes wrong, like mutation.